I think this is working right now. I hope that it is. I'll watch back. Note to editor: watch the video again to see if there's audio actually working. But anyway, what's up, y'all? What's up, y'all? Or actually, hello, Dr. Tice. I hope the subtitles spell your name correctly. Yay, it does. Um, but it's Calvin, and we're gonna be talking about plants and nutrition. This will be kind of like this will be structured similar to your lecture, because I'm not really good at this stuff or at talking about stuff. But yeah. But initially, I wasn't going to be talking about stuff. I wasn't going to be doing a little presentation. But I just don't have a time to do other stuff. I did think about doing a rap video about the Calvin cycle, but I am not going to do all of that. I'm not going to do all of that. Why, you may ask? Number one, I'm not a rapper. Number two, I don't have the time to become one. And also, I just have too many finals coming up. I have a lot of finals coming up, or I have quite dense finals. Let's just say that I have quite dense finals that how do you say, demand a lot of attention, demand a lot of studying. Which so, I just don't really have the ability to make a rap video, and I feel like I'm not really good of a writer when it comes to rap videos and stuff like that. So, yeah. Um, yeah, so I'm not gonna do all of that, but I hope you enjoy this rest of the thing anyway. So let's keep on going, right? Shall we? Bang! Autotropism. So we typically like to eat things to survive, right? We depend on biotic material and we convert it to nutrients for our survival. Plants are really cool because they take abiotic sources of energy, so non-living sources of energy, and convert it to organic compounds that are important for uh, growth and maintenance of said organism. And I have some couple of examples here. The classic example of autotropism is the plant. The plant takes in sunlight, carbon dioxide, and water, none of which are living things. They are abiotic. And they convert it to oxygen and sugars. Again, these are both abiotic materials, but they are used for survival. Sugars, sugars is used for overall maintenance of the plant for the plant to grow, and oxygen is overall just waste material for the plant. And there are also some examples like plankton and algae. And the cool thing about it, I learned that plankton need light. They need light to survive. Oh, why did it move by itself? No, no, don't do that. But I was talking about plankton and SpongeBob, at least SpongeBob. I love SpongeBob. When you see plankton eating this sandwich thingy, is that sandwich or is that turkey? I don't know. But the sandwich is actually not a sandwich. Crazy, right? It was a hologram, and that's where I learned plankton need light. They photosynthesize, apparently. I guess they photosynthesize. I don't know what they do. But yeah, cool. Um, um, and the next thing what we have here is how about do plants use the abiotic factors of sunlight, carbon dioxide, and water to make sugars? Well, they do this fancy thing called photosynthesis, as alluded to before. Here's the overall equation of the photosynthesis. You have 6 moles of carbon dioxide combined with water, and that yields glucose and 6 moles of oxygen gas. Photosynthesis happens in 2 separate processes called the light dependent cycle and the Calvin cycle. And you see my little face right here. That'll be a recurring theme throughout the slideshow, because I just took that selfie 5 minutes ago in the bathroom. But in the chloroplast, plants take in water, and the water gets split. The water gets split by the energy of the sun into oxygen and hydrogen. And the hydrogen gets the energy released from that breaking up of electrons and hydrogen gets fixed into ATP and NADPH, which then gets used in the Calvin cycle to convert carbon dioxide and fixes it into glucose. And then of course NAD plus and ADP gets sent back to the thylakoid to overgo this process again. So this harmonious process that we call photosynthesis actually consists of two parts: light dependent and Calvin cycle. And the Calvin cycle can be summarized by this graph. We're not going to go over it too much, but the most important things to note that is that the Calvin cycle from this is a lore from Biology 1 happens in 3 separate stages. But we'll be mostly going to be talking about stage 1 and Rubisco. Rubisco is the enzyme that fixes carbon dioxide into these intermediates, which then gets fixed to 3-PGA. And with the use of ATP throughout the cycle, it gets converted to a glucose, and the glucose which is sent to the cytoplasm, but some of it gets sent back here, and gets fixed to RUPB, which then gets fixed by Rubisco, and it goes through this constant cycle. It goes through this constant cycle of carbon dioxide to 3-PGA to glucose, and so on and so on, which makes nutrition for the plant. And you might be wondering how about do plants carry water and nutrients to and fro the plant? Oh, and it just changed at the perfect time. We have 2 different types of tissues that plants use to move water and nutrients around: the xylem and the phloem. The xylem is one-way flow of water from the roots to the shoots, and phloem is 2-way flow. You can it's water and sugars moving to and fro wherever it needs to get moved, to plants, to flowers, or wherever. It moves in both. It can move in both directions, up and down. How does xylem move water up? It's basically through a motion of osmosis. Osmosis is a passive process where water moves up from a place of high water potential gradient to a lower water potential gradient. It moves up, and thanks to the forces of cohesion and adhesion, water will stick. Adhesion is where water will stick into substances, and water sticks. Water will stick to the xylem and move up. Oh, that's not what I wanted to show you yet. But sorry for the technical difficulty. But if you take a look here, water will move up from negative water potential. Negative water potential will actually drive water to the root and will push it up. And it goes, and the final destination of water is typically to the leaves, where it gets released by transpiration. And whatnot. So it all happens thanks to the process of osmosis. And you might be wondering, isn't osmosis just diffusion, but it's water or whatnot? Well, it's actually just more than that. It's not the full story. Yes, it is water diffusing, because as you know, diffusion is a passive process where water or any other liquid or any other particles, they move from an area of higher concentration to low concentration. So this is solvent stuff. Why does my stupid slideshow keep moving automatically? I don't know why it does that. I don't know why it does that, but you're gonna have to deal with it. But anyway, through a semi-permeable membrane, water will move to and fro an area of high solute concentration to low solute concentration, and the basic idea is it moves to make the concentration of overall things consistent throughout. And when you have non-water particles, so solute particles, that increases the pressure, that changes the pressure of water, which forces water to move passively. This is pressure driven, not energy driven. So there's passive transport. And we measure this with typically with millimicropascals, as we have higher solute potential to lower solute potential, or a lower water potential to a higher water potential. Water gets pushed passively to the to an area of low solute concentration to high solute concentration, in order to increase the concentration of not increase the concentration, but to keep the concentrations consistent throughout. And this is more of a physics thing, so I'm not really well too versed with it. But you notice that, remember, we're moving from an area of higher potential to lower potential, and notice the negative MP3s, not MP3s, MPAs, which is micropascals, and we have a little bit of the same notion in here, where we have solute concentration, and water gets pushed up with the water gradient, because remember, at the atmosphere, and it moves up to the atmosphere, and the atmosphere has negative 100 micropascals. That might be megapascals, actually. Sorry about that, but yeah, it's mega because it's a capital M. I think it's milli. It's in millipascals if it's lowercase m. I don't know, but yeah. The main concept is higher potential to lower potential of solute, but it's lower water potential to high water potential in here, right? Or high water to low water potential right here. Sorry about the switch up. Negative water potential will drive the water particles to the roots. All right, and then phloem kind of moves in an interesting way. Phloem can move to and fro depending on where are the nutrients needed at that time. And this is through a motion of active transport. Active transport is using ATP to move things around. And the cool thing is, as water moves up, remember, water moves up, we have source cells. We have source cells that have sucrose. They have sucrose, which of course reduces the water potential, which makes water rush into the sources. Which and notice when water gets pushed to the source cells, we have pressure. We have pressure changing in the sieve tubes, which forces water out and forces sucrose out in consequence, and it pushes down through a sink cell. Source cells move into sink cells. And these source cells can be seen in the. I think there's another way of showing these off. Companion cells, and these companion cells use ATP and amino acids to force sucrose into these little plates, which then the internal pressure pushes water down, that's how water is able to push into the direction that it wants. And it doesn't always move down. It can actually move in multiple directions. Source to sink is the main importance here. So there's a little example. I might not go through this too much. It might store stuff in roots, so like tap roots, for instance, where it stores carbohydrates under the ground. Also, during the summer, where lots of photosynthesis is happening, but in times where there's less resources, where there's less water, or there's less of sunlight, more of the nutrients will move up from the plant to where to the leaves or whatnot, or to the shoots where the nutrients are needed. All right, let's keep on going. More on the Calvin cycle. We're back to the Calvin cycle. The reason why I wanted to discuss this Calvin cycle is because of this little thing, Rubisco. And Rubisco's main goal is to take in carbon dioxide and fix it and to do all of this stuff. So the Calvin cycle can actually happen, and we get some proper glucose, right? It's quite efficient in the way it does that. The problem is Rubisco is not designed to just take carbon dioxide. It has an affinity with oxygen. Remember, oxygen is also small. Oxygen is also smaller. smaller than carbon dioxide. So it is most likely to take in oxygen in these sites as well, and which leads into photorespiration, which is actually a waste of time. Oh, I don't know why I skipped to that. All right, I didn't mean to do that. Sorry, but it makes it to waste product. And you might be wondering, why did it evolve that way? And my accidental switching of the slides kind of alluded to that. Is because when Rubisco was evolved, it was useful at the time. At the time, we were at a time in the very beginning of the Earth where cells, where there were unicellular organisms in the water. They were absorbing a lot of carbon dioxide, and there was a lot of carbon dioxide back then. There was not much oxygen then, but because these cells had practically an infinite food glitch, and they evolved this thing called photosynthesis, and they were constantly photosynthesizing. It became so that oxygenation of the atmosphere started to happen, but Rubisco did not make the proper changes. Rubisco did not make the proper changes to prevent oxygen fixation, but it got so complex over time that it was really hard to come back. It was really hard to come back. So we were on a notion like there's way more oxygen in the air than carbon dioxide is, and that suck. It does suck, but fortunately, in ideal conditions, this doesn't serve as a big problem. Introduce here. We have photorespiration in the Calvin cycle. And we have Rubisco. And this just shows the basic processes right here. And those in photorespiration, you do make while you do make waste, you also use more energy. You also use more energy here and whatnot. But also you get this notion of like the materials that you make must be used to make the Calvin cycle. So you have this you practically. You get away from the Calvin cycle, but you get back to it. It's kind of a waste of time for plants to want to do that. And okay, this is finally alluding to what I want to talk about is, it's becoming a big problem for plants to deal with Rubisco because of this climate change or global warming. And a lot of people, I know there are some people who are skeptical about it. And one of the main arguments is, but doesn't the Earth's temperature change? Like, isn't like we've seen the ice ages, or we've seen like the Permian extinction, where the temperatures of the Earth moved and shifted to and fro, high to low, and over time. Like, isn't it not a bad thing? Well, I'm not saying that changing is a bad thing, but the rate of change is kind of concerning. Here's a little figure of the global temperatures over time, and so this is the lines represent the average, at least our predicted average, and the blue line represents what we've been able to record. And you notice that this kind of change started in around the 1800s and the 1900s, and that, I'm not going to say anything, but that coincides when oil was discovered. Remember this oil was found to be underground in the 1800s. In Galveston, Texas, oil was found in the year 1900, which is how it boomed to this crazy economy of oil. But as oil became used for energy and plastics and all those things, we've become dependent on oil at a cost of the Earth's overall equilibrium temperature. As we notice that over the times, while it's fluctuated, it was able to finally control itself. We have reached a point where, unless we make some really drastic changes, it's going to be a while. It's going to be a while until it can probably go back to equilibrium, and it probably won't. And that's a problem because photosynthesis happens. Photosynthesis happens at an optimal rate. If it gets too hot, then the proteins, i.e. Rubisco, will actually deform and degenerate, because proteins can work only on certain pHs and certain temperatures. So photosynthesis cannot even happen when there's too high of a temperature. So how do plants get around it? Well, this is our usual C3 pathway, where we have in the mesophyll cell, we have the Calvin cycle happening at the same location where we have CO2, opening via the stomata, gas exchange, and sugars are made in the same site. But with climate change going on, with heat, with hotter temperatures going on, opening of stomata risks loss of water, and also we risk more oxygen getting up in the air. So that's actually a big problem for plants. Because of photorespiration, and that leads to inefficiency and just loss of net energy overall, because you're spending more time trying to use more energy to convert the waste product into useful sugars. That's just a net negative. So plants have thought of these adaptations in times of extreme temperatures to still do the Calvin cycle. And there are still trade-offs with temperature, but these have worked for plants in times of heat, in times of the stress, times of resource scarcity. Let's take a look. C4 pathway. C4 happens in spatial separation, and these happen through bundle sheets. So it's a physical wall that separates, While we have CO2 here, CO2 gets fixed into CO2 gas exchange gets fixed into oxaloacetate and malate in the mesophyll cells, and then gets converted into carbon dioxide, which can be used by rubisco, and this gives rubisco more opportunities to get binded to by carbon dioxide, and less opportunity for rubisco to get binded by oxygen, because it's literally getting blocked off from the outside world, right? And the Calvin cycle happens here, and sugars gets made, and then pyruvates get sent, and then pyruvates get sent back, and the pyruvates gets fixed with carbon dioxide to make oxaloacetate and malate, and the cycle happens over and over again. So it's just a longer version of the Calvin cycle. And yes, while there is trade-offs that can be made, when you compare the trade-offs in times of extreme temperature, the trade-off becomes worth it. It becomes worth it. So yeah, there's another thing called CAM, where we have stomata opening and closing depending on the time of day. So that you don't have much water loss, because water is an important thing to it as well. Without water, you cannot do light-dependent reactions. You can't have the energy needed for the Calvin cycle. You don't have the ATP and NADPH without water. You need the water. So what happens here? So in the night, stomata gets open for CO2, and then of course it gets converted to oxaloacetate into malate, into organic acids, and that stuff gets stored at night. In the daytime, while the light dependent reactions are able to happen from the side, so you see, imagine a separate thing where we have water getting converted by the thylakoid into oxygen gas and hydrogen ions, which then gets fixed into the ATP and NADPH, which then is used for the Calvin cycle, and was again a longer version of the Calvin cycle, but in times of stress and in times of change, that stuff becomes worth it in the long run. So yeah, that is how plants have adapted to get nutrition. And I do think that is the end of it right here. That is it. I thank you for listening to everything I have to say. I hope I've reached 5 minutes. I think I have. But yeah, that is it from me. I'll see you on Tuesday, I guess. See ya. Bye-bye. See, how do you stop the recording? Oh, there's the button. There it is. Stop screen recording. Left click.